Right, thank you very much for the kind invitation, for all the good work at SwissCollNet, and it's really wonderful to be here today. So today I'm talking about data standards in practice, and I'm going to start a little while back, namely about 401 years ago. So 400 years ago, the first floristic work from Switzerland appeared, which was a local flora of Basel and its surrounding areas, which was published by Caspar Bauhin. And the format may be unfamiliar, but we have a name, Lilium floribus reflexis montanum. Then there is some synonyms, and then there is a locality in Monte Muteto at Grenzach. So if you're from Basel, you know where it is more or less. So this is, it contains all the information. Now, local floras are generally very interesting because they anchor plant diversity in space and time. So it allows you to know what grows where and under what name can we recognize it. But it is more than trivial to figure out what is the good name, especially 400 years ago when there was no clear databases and no clear sort of standards on how to do things. And that means that the development of these local floras was in association with the development of complete compendia of plants. So if you want to know which plants grow here, you need to know what it is and what it is not. So that is the Pinax, which is 400 years old. That's this book right this, which is a complete overview of all the plants that you have. Now, Bauhin was an early botanist, but he was not the only botanist at the time. So that means if somebody in Germany had written a book saying, I see this and this species here, Bauhin would like to know, what is that species? So he sends letters and asks, send me a plant, please. I need this because I want to know what it is. So that means developing these compendia that are necessary for floras requires exchange of plants and ideas, correspondence network, etc. So this is the origin of voucher specimens for names. Interestingly, the word specimen didn't exist yet. That's a word that was invented in the 17th century, but the practice was already the same. So this is the early origin of Herbaria. And in Basel, we're lucky enough that one of these very early Herbaria has survived, and that is the Herbarium of Caspar Bauhin, of which you see about 9% of the specimens here. So it's assembled from the 1570s to the 1620s, more or less. Today, we have about 400 million herbarium specimens in the world, and I want to draw your attention to this graph. I'm pointing at the left thing here. There's this tiny little red dot. So this is the year that Herbaria were founded on the database of Herbaria, and you see that Bauhin's herbarium was really at the beginning of botany. First flora of Switzerland was still at the beginning of botany, and only in the 19th century Herbaria were arising. That means that at some point you had standard specimens, and we know how to deal with these things very well. And that is the absolute bulk of specimens, say the 99%. But then you have the 1%, and those are the really old and not so standard specimens. Yet, we want to make them discoverable, accessible, so we need to apply data standards developed with those modern specimens in mind, sort of retrospectively on these older specimens. And that's a bit of a challenge. So what is a specimen? A specimen is a thing representing a single gathering in space and time. So it's usually, in this case, it's a plant, and there's a label. And these things are placed into boxes, and you know where to find one if you know what species you're looking for because of a filing system. But there's no interaction between the objects, so to say. All the information is contained on the specimen. It's visible on the label. If there's interpretation necessary, as Holger pointed out, it's fairly straightforward. And that also means that the digitalization process is sort of a one-by-one step. You do one specimen, then you do the next, and the third. And you can do them in any order and in any sort of set. And this is for which Darwin core specimens were developed. Now, I'll only sort of briefly mention this. A typical Darwin core entry for this specimen, I'm sorry if it didn't really show up very nicely. This is the label of this specimen. So we have a species name. It says Flora helvetica. There's a Wäldchen auf Blockschutthalde unter Felsband in Wallis Lagintal, west of Wechsel. It was collected at a date at a certain elevation by a certain person. So we have information on this specimen at the record level, at the occurrence level, at the taxon level. There's an identification. There's an event that happened, the gathering, etc. And then there's location information. And most of this is either verbatim information, or it is so easy to interpret it that you forget that it's actually not verbatim. For instance, the species name, Primula halleri, we write it with a small capital H now, and the author abbreviation is standardized in a slightly different way. Other fields require slightly more interpretation, like for instance georeferencing. If we look today, in the Lagintaal, there is no wegsel, because the name of the locality has changed in the 70s, because the spelling changed. So looking at old maps, it's fairly easy to pinpoint the specimen to within about 100 meters. So all fields except the real verbatim fields require some sort of straightforward interpretation, and Darwin Core captures both the verbatim and interpreted data. So classic collections of these standard specimens are sort of unstructured elements. There's no intrinsic association among elements beyond the filing system. These are primulas, so they go in one box. These are orchids, they go in another box. Simple as that. And it also means that the scientific value of one specimen does not depend on the content of other specimens. That sounds very esoteric and irrelevant and obvious, but it becomes obvious in a minute and important. So if these specimens, I lost the dot, doesn't matter. So if these early specimens are so rare, and so why bother with them anyways? And that is because I think we're sort of at the beginning of the second renaissance of herbarium-based research due to technological advances. So it is nowadays, but only recently, possible to generate complete ancient plant genomes from old specimens. And that means that due to this, the early specimens, the earliest specimens, become more and more important to be findable, and not just by the curator, but by other researchers as well that are interested in using these things. For instance, if I get a request for which of these 10 species do you have specimens at least from the 16th and the 18th century, I need a database to query that quickly. If I don't have a database and these things are not filed properly, I cannot get at that kind of information. So now I'm going to give a couple of examples of these early specimens and how to interpret them. In some ways, it's fairly easy. Like, for instance, this specimen of Caspar Bauhin. It says there's a name, Solanum pomiferum fructo mollis, which is an old name for tomato. This is indeed a tomato plant. There's some synonyms, and then it says Basilea ex horto. That means Basel from the garden. So by reading this, we can interpret it as the provenance is from Basel, the garden of Caspar Bauhin, which means it was collected after that garden was established and before he was dead. So that gives a range. And we use the field event date to capture a range for the collection. And we still enter the verbatim information as well. There's other specimens that are more difficult to interpret because there's no information immediately on the specimen, and all the information comes from the context of the specimen. This is also a tomato. Half of this leaf has been used to generate a complete genome sequence for this very specimen. And it comes from a collection, 5 boxes that were neglected because Beans in 1908 claimed, der wissenschaftliche Wert dieses Herbariums ist übrigens gering. Da Fundortsangaben oft fehlen, etc., etc. And then because it's so beautiful, viele der darin enthaltenen Pflanzen sind auf Papier sorgfältig aufgeklebt, mit später ausgeschnitten und mit wunderlichen aus Kräuterbüchern entnommenen Namen versehen. Vermutlich sind diese Stücke aus noch einem älteren Herbarium eingereiht worden. And if you look at the specimen closely, it is wunderlich, it is remarkable, because these are not fruits. These are the skins of fruits that were then cut and glued to the paper. Also, these are flowers and those are fruits, and tomatoes do not tend to flower and make fruits at the same time. Right? Then this is not a stem. This is a stem, and these are leaves that were just sort of glued at sort of nicely looking places at the specimen. So it's a curious specimen, and if you look at it, it is reminiscent of the herbarium of Felix Platter, who was the teacher of Bauhin, who was a medicine professor in Basel, who did something similar. So might this be a specimen of Platter? Let's look at that a little bit. Looking further at the collection, there's other specimens that are like this, and many have little corrections on the labels. We can look at the paper beyond the, because this is only a name. There's no further information. It's an old name. And there's a correction on the label. If we look at the quality of the paper, we can look at the watermarks. And we see that all the paper comes from Basel. You see the Basler Stab here in what is referred to as a Lorbeerkrans, so a lauraceous thing around it. And these little symbols that you can see, and based on this information, we could know that this paper was produced in the 1730s in Basel. The plants must be younger than that because they're glued onto this paper. This information does not come from the watermark in the tomato plant, it comes from the watermark in the other specimens. Moreover, we didn't know who collected it, so we looked at a lot of labels. And without going into detail, the abbreviation arises very often in combination of the year 1759, 1760. This is Leiden, so somebody was in Leiden. He was in Leiden in this time. And among the people that are mentioned on the labels, a couple of people are called friends. So connecting this, checking the matricle of the University of Leiden, these three people are exactly the three Swiss people that were immatriculated at the University of Leiden in 1759. And in fact, there's a fourth person. And that is the only person from Basel, and that is Johannes Ludwig Buxtorf. And then, so that means because of the collection context of the specimen, and via this information, who he called friends, figured out who collected these specimens. Unfortunately, we still don't know who collected this very specimen, so in terms of Darwin core, it becomes simple. Collector is unknown, and the event date is, well, let's say 1720, because the dating of the paper isn't entirely accurate, and 1750, because it would be unlikely that paper would be around for so long. Certainly not from Platter, and this is for the genomic study very important to know. However, in this collection of Buxtorf, there are many, many other specimens, and in fact typical, a single sort of specimen looks like this. What do we have? We have two plants. We have one here. We have a drawing that's annotated with something. And we have another mounted plant with a label with corrections in the same handwriting. On the front side of this folio paper, you also see a correction. So this is one thing that contains other things. It's one folder that are things that are clearly associated, one species, but it contains other things. So how do we think about it? What is the specimen in this context? So if you ask archives, I don't know if you call it archivologists or so, people that deal with cataloging the inheritance of people or so, there's a difference between a specimen and an object that we tend to forget because they're the same with modern specimens. A specimen is a physical object that has been collected for scientific purposes, typically study or analysis. So that means the intent of the collector makes something a specimen. And that would be a plant and not preserved, things that are not preserved in the specimen. An object is a physical item or material that's being preserved for its historical culture or scientific purposes. So we as curators make things into objects. It's not the collectors that make objects, we make objects. It also means that objects are more inclusive than specimens because they contain information on the paper and how it's assembled, the physical state, etc. And in this case of the Buxtorf herbarium, it's that physical state that leads us to interpret the specimens. So what is then the collector's intent and what was it? Modern intent of specimens to document a single occurrence of one species, of one organism at one point in space and time. And the label contains that information on the occurrence. But this is not the way it was in the 18th century. The historic intent was a typological representation of a taxon, which features more, on which you can see more details than on an illustration. So that is why the labels contain multiple provenances. The specimens contain multiple plants from different sources. There's multiple labels because they were received from multiple people, etc. And this shift in concept happened in sort of the second half of the 18th century slowly. So that means that the intent of that collection of things, of items, was different from how we now want to catalog these things. So that means there's consequences for specimen interpretation. So this is the same thing again. One thing with multiple things, hierarchical structure within the specimen, and one thing, no hierarchical structure. So the standard object and the herbarium specimen are the same, typically in collections, but not in the older ones. That's all I want to say. So how to think about these things? And then coming back to the science of archiving again, we think of these things at various levels of aggregation. So if you think of an inheritance that you're describing in a catalog, there's also a hierarchy to something. There's a cupboard and there were things in the cupboard. The levels of the hierarchy that are used are record group or collection, series, file units, and items. And an item is the smallest intellectually indivisible item. In this case, for this kind of historic specimens, I would consider this an item, this is an item, this is an item, this is an item, etc. But only one of them, this one, is actually a physical object and a preserved specimen in the sense of Darwin core. Because this one is the one that contains the occurrence, habitat in fossis Michelfelde, that's written here. Now these things are still physical objects, and hopefully it's obvious that we want to preserve these things and database them as well, but they're actually not preserved specimens because there's no plant associated with it, because there's just a bunch of plants that all go with this label. So that means as far as I know, there's no basis of record class or vocabulary that applies in this case. So we're using the word label for this, or naked label. And there are relations among the items within a file unit, and those are given by parent-child relations, which you also use if you have a plant that is mounted over multiple sheets. One is the boss and the other one are the children. So, and the standard label, this is the parent because it contains the occurrence. Okay, so complicated story. What would I like you to remember? That is, things become specimens because a collector wants to represent something. Things become objects because a curator wants to preserve it. Historic specimens require careful interpretation in their context, so that means it's our challenge to also preserve that context. And not every item that is preserved and cataloged is actually a preserved specimen. So we catalog and preserve things that are not preserved specimens as well, such as those labels. All of this, I'd like to stress, is thoughts in process. I hope that I get lots of comments and questions and people telling me that I'm doing it all wrong. And I'm looking forward to the discussions later. Thank you very much.
Thank you very much, Jurriaan, for this inspiring talk. Cool. Any questions for Jurriaan? Michelle?
Yeah, thanks, Jurriaan. Well, I don't know whether this is working or not, but thank you very much for the talk. And it's sort of a question after what you're doing right now is, how do you assign a unique identifier to all this? Because obviously, if you want a barcode on this specimen, you have to be very careful about to which specimen you link it, and do you link it to the folder and have other ones? I just wonder if you've thought about that, how you use your identifiers on this type of stuff.
Yeah, I thought about that. So in general, every item needs a unique identifier because those are the things that also we want to tally. We can identify the handwritings. For instance, we want to know how many specimens of Gagnebin are within this collection, and they will be part of these folders. So they need, those are the sort of the granularity that we need. What I don't know yet, if we are assigning a barcode to the whole folder as well, like in addition to the items in between. So far, I'm thinking to not do that, but assign one as the parent and the rest of the children of the parent. There's another practical problem. Barcodes are not tiny, and even if they are small, I don't want to stick things on these. That would be helpful. Alternative is to actually make a digital image of a specimen like that, and then have sort of a legend associated with the specimen that would say, this is this barcode, this is this barcode, this is that barcode.
I'm also dealing with historic specimens of later than this, and I really was against sticking the barcode onto this historic specimen. So my solution was sticking it onto the folder.
Yeah.
Of course, this is not ideal because if somebody swaps a folder with a different specimen, it's not there. But again, it was a little bit, I don't have a problem sticking it on something from 1973, but from something from 1700s,
Yeah.
I got a little...
So the idea right now is, this is a folio thing. I opened it, made an ugly photo. So to use for these specimens, sort of type folders like things, and each type folder contains two things. It contains the actual folder with the objects or with the items, and it contains a photo of that, in which contains the barcodes, and that says this is this, that is that.
Thank you. Another question for Jurriaan. Celia?
Hi, just thank you very much for the presentation. I had a question. How do you register and encode the information in the items that are handwritten?
Yeah. So what I'm not going to do is transcribe everything, like every single word. But luckily, these labels are very structured in a specific way. There's a species, if it's linear or not, and then there is something where the species occurs, sometimes with an addition saying I got it from this and that. So it's a statement on the species and then where he got it from. And sometimes it says, rather frequent, from whom he received the specimen. So those are the things that we would register. What occurrences are listed on the species and what persons are mentioned. The question is still, which I haven't solved, maybe somebody has a good idea. There's also things that say, ex herbario vivo doctori Stehelini. So who is the collector then? Is it Buxtorf because he made it into a specimen and entered it in his collection? Or is it Stehelin because he killed the plant and gave it to Buxtorf? So far, I'm saying that the collector is Buxtorf, but I know that is not, it's subject to interpretation. So if somebody has a strong and good opinion about that, I'd be looking forward to hearing it.
One last question for Jurriaan. I have a question myself. So how does this translate into your database when you have a lot with LOT with multiple items into it?
So what you would search for is, I mean, it's always one species. The one folder is always one species. But you would, so we are using the Digitalis database that was developed in Zurich. But we're still in the process of rolling it out. But in principle, you can search for the different collectors or the different persons mentioned, and you would find it. If you would find but it would appear as one record for the whole thing because of the parent-child relations. So you only have the parent is the specimen that appears, even though it can contain multiple occurrences, etc.
Okay, thank you, Jurriaan. Thanks again for your super talk.